We've been working so long together, well, actually, we've been working for about 2 years together on a project on genomic sequencing and newborn screening disorders that, we've become partners in crime here. So we're going to tag team our talk. So I just wanted to give that overview. So Anastasia, go.
Okay. So we just wanted to start off discussing a little bit about this newborn initiative that we've been working on jointly between NICHD and NHGRI. And this really came out of the fact that initially when we started talking about human genome sequencing, it took years and years to sequence the first human genome. But now, today, we're really talking about sequencing thousands of individuals in a time frame more along weeks or days, and the cost has really dropped. So today, sequencing the genome is starting to get to the point of being able to be practical in a clinical situation, and that causes us to really have to think about, is this something that we want to be doing, and in what situations lend themselves best to doing clinical genomic sequencing?
So, as all the speakers have let us know today, there's a tsunami of information out there. There's many problems. There are a lot of issues. How are we going to all deal with that? That's the one big question. People seem open to wanting to deal with it, but what and how are we going to deal with it? So to prepare for these changes and the impact that it might have on newborn screening and child health, Alan Guttmacher, the director of the NICHD, and Eric Green from NHGRI brought together staff members with the intention of organizing a workshop to discuss these concerns, because one of the things that was coming up was, this is going to happen, and people would say, well, we're not ready, or how do we do it, or what should we do? And our perspective on this, or the director's perspective on this was, if this is happening, we need to address it now, whether we like it or not. Something needs to be done. We need to be looking at it. So both of the institutes came together, and we sponsored a meeting with a very glowing baby, the newborn screening in the genomic area, setting a research agenda. The purpose of the meeting was to identify elements of trans-NIH research agenda that would lead to applications of new genomic concepts and technologies to newborn screening and child health. So it's really a partnership of the two institutes. The meeting was attended by experts from academia, industry, the federal agencies in the fields of newborn screening and genomics. So it's two groups that really don't naturally come together necessarily. The meeting was chaired by David Valley, who represented the genomic side, and Piero Rinaldo, who represented the newborn screening side. And there's documentation of this meeting, a meeting summary, if you just Google newborn screening in the genomic era, and there's that if you get the slides. The real agenda for the meeting was to set the research agenda and find some potential first adopters. Really look around at who's interested in doing this, who's interested in answering some of these questions. This is a situation where we have near universal inclusion. We are screening millions of babies, and this could lead to a lifetime of personalized medicine. How are we going to handle it? How are we going to manage it? And one of the first things we wanted to do was set up pilot studies to examine the situation. And one of the big debates that came up in the meeting was newborn screening, the current public health program, versus the screening of newborns, using sequencing to screen newborns for multiple conditions. And that's also been discussed a little bit. What environment will these kids be screened in? When is the right time? How to do it? So the primary outcome of the proceedings was the importance to evaluate genomic data in newborns using newborn screening as a framework. The second was the importance to prioritize clinical validity and clinical utility, not just analytic validity. And we wanted to look at the importance to address the ethical, legal, and social concerns. One of the things that came up was people were saying, well, we should look at the ELSI concerns first and then look at the other parts. But our sense was you need to look at it in a real-life situation, all working together. And from this came our three-legged stool of our initiative that some people are hoping and chomping at the bit to find the answer as to who won the awards. But that information will be coming out at the end of the month and the beginning of next week, hopefully. So this was genomic sequencing in the newborn screening disorders, and we received wonderful applications for this. The research community really rose to the occasion, and put together some nice applications. So for those few of you who don't know what this was about, we put out the initiative, and we wanted the research community to answer some questions. For the disorders currently screened for in newborns, how can genomic sequencing replicate or augment known newborn screening results? What knowledge about conditions not currently screened for in newborns could genomic sequencing of newborns provide? And what additional clinical information could be learned from genomic sequencing relevant to the clinical care of newborns? We really wanted to look at this problem in a very thoughtful manner. The projects each needed to include an ethical, legal, and social component, genetic data sets, or genomic data sets, and a clinical research component. And Anastasia.
And we did also want to inform you that along with those U19 awards that we'll be making shortly, there is a second program that's related to this newborn initiative that's for small businesses, related to developing methods for obtaining comprehensive genomic information from human specimens that are easy to collect and store, which is a long way of saying that we're looking for other ways of collecting samples for doing things like newborn screening in a way that you could have a sample that you could easily store somewhere, that you could collect in a more non-invasive or minimally invasive setting, and you'd be able to get from that sample a lot of genomic data. So for this small business or PAR, we're asking for folks to develop sensitive technologies or methods that obtain high quality and comprehensive genomic data from limited quantities of human specimens that are, once again, that easy to collect, handle, and store phrase. So for this, we really are looking for genomic data. So they can't just be used for doing small amounts of testing, and they need to be able to do genomic sequencing or other such methods from the samples. Overall, there are a couple of specific review criteria to this PAR. The first of which is that the ease of sample collection needs to be at least comparable to the current dried blood spot collection method. So it needs to be at least as easy as a heel stick, though I think for the baby they probably don't particularly like that method. And then on the other side, it needs to be able to obtain genomic data that's at least equivalent to what we can get from whole blood in both its quality and its comprehensiveness. So we really want to make sure that these are technologies and methods that are advancing from the point that we already have today. For these small business awards, we're accepting applications for Phase 1, Phase 2, and Fast Track awards, which combine both Phase 1 and Phase 2. We've received the first batch of these applications, and the next due date is in December, and there will continue to be deadlines March, July, and December from now through March of 2016. And overall, the funding support will be up to $225,000 for the Phase 1 6-month awards and up to $1.5 million for the Phase 2 2-year awards. We also wanted to share with you a couple of other resources that are not necessarily directly related to newborn initiatives at the NIH, but some of the other things that might be related at both NICHD and NHGRI.
So as many of you know, Newborn Screening Saves Lives Act of 2007 has really was able to kick off a lot of the initiatives that we have at the NIH. We have the Hunter Kelly Newborn Screening Research Program at the NIH, and the goals of the program are projects that identify, develop, and test the most promising new screening technologies. They increase the specificity of newborn screening and expand the number of conditions for which screening tests are available, and develop experimental treatments and disease management strategies for additional newborn conditions and other genetic, metabolic, hormonal, and functional conditions that can be detected through newborn screening for which treatment is not yet available. So people often come to us and look for a specific initiative and say, well, you know, I can't find an initiative on the NIH website that's directed towards newborn screening, but if you look at this initiative, you know, we basically have legislation telling us what type of research we need to be doing. So we're always looking for investigator-initiated projects that are related to any of these points. We are also funding the Newborn Screening Translational Research Network, which the primary aim is to develop a research infrastructure to support investigators with projects related to newborn screening. It was initially awarded in September 2008 to the American College of Medical Genetics. It was a 5-year contract. It's currently being recompeted, and it should be awarded by the end of this fiscal year, which is the end of September. Part of the MBSTRN, we have the R4S, which, don't ask me what that stands for, but I'm sure many of you can tell me. Region 4 Stork, but that won't mean anything to you. But the analytic and clinical validation and laboratory protocols and definitions are part of this, where state laboratories can put their data in and do comparisons. And Como can maybe speak to this a little bit if you have any questions about it. They use this a lot for the SCID data. There's a virtual repository of dried blood spots, which is sort of, I think of it as the Amazon.com of finding the dried blood spots you need, in that the states own the dried blood spots and control who they go to and who they don't go to, but the virtual repository helps investigators find them. So instead of having to go to 50 different states and talk to 50 different people, this is one site that can help you go find the dried blood spots you need with the states that are participating. And there's also the LPDR, the Longitudinal Pediatric Data Resource, which is secure standard-based clinical data collection and management. And we allow investigators to put their data in there. We work with them to do long-term follow-up of kids. We right now have some pilot projects going on, one with spinal muscular atrophy and another using many different disorders. They also are adding a genetic component to this to be able to collect the genomic data into the data set. We have the genetic test registry, which is fairly new at the NIH, replacing an old system, but this is a place where clinicians can find tests and find information about the test. It contains phenotypes, looks at the genes in the labs. You can find information about the genetic tests and the genetic disorders. And one thing that's nice is if you're developing some new tests, you can also post them there so you can have a research type test, one that's not standardized yet. So.
And we also want to talk a little bit about some of the other sequencing programs we have that are looking at the genomic sequencing in some other areas other than the newborn screening arena. And in the Undiagnosed Diseases Network, which is an NIH Common Fund program, we're looking to take the program that currently exists at the NIH Clinical Center for looking at undiagnosed diseases and expand it to a network of clinical sites across the country that would make these services more available to the general public, as well as work on creating a set of protocols that could help with developing management strategies and protocols for assessing patients with undiagnosed disorders. Overall, we're starting with the current existing undiagnosed diseases program, and we've put out solicitations for a series of clinical sites as well as a coordinating center for this larger undiagnosed diseases network. We currently have applications in for some gene function studies, and these studies will continue to be solicited throughout the duration of the Undiagnosed Diseases Network. These first couple of solicitations were based off of variants that were found in patients seen at the clinical center at the NIH, but as the program progresses, we'll add new variants that are found from all of the different clinical sites, and there will be continuing solicitations for folks to follow up on these and do continuing research to determine what the function is of the variants that are found in these patients, as well as a DNA sequencing core, which we currently have a solicitation out for now that's open to find a DNA sequencing core that will be able to provide clinical sequencing for all of the patients seen through the NIH Undiagnosed Diseases Network. We're looking for this center to be able to provide DNA sequencing as well as CLIA variant validation for all of the patients seen, as well as some of their family members, and provide raw sequence data back to the clinical sites within at most a 2-week turnaround time. So we really are looking for folks to push the envelope to be able to get this clinical data back in a shorter time frame in order for it to be more usable in a clinical setting. And then finally, I want to talk just a little bit about our Centers for Mendelian Genomics, which are also looking at sequencing, but in this case, rather than sequencing for a diagnostic purpose, they're sequencing individuals that have known conditions in order to discover the genes and variants that underlie those Mendelian conditions. And the Centers for Mendelian Genomics do accept samples and families from researchers outside of the 3 centers. So if you're interested in working with any of them, I've provided some of the contact information here. You can contact them, and then the centers themselves go through a selection process to determine if the the samples will be placed into the queue for sequencing at the centers. But even if these samples are not selected for being sequenced as part of the Centers for Mendelian Genomics, they can also be put into a repository that they're housing in order to collect samples from multiple different Mendelian conditions, and then potentially could be reassessed at a later time if more samples were made available in order to provide a larger cohort for sequencing. And with that, I think we will end. We just want to give you a brief overview of some of the different programs that we have at NICHD and NHGRI, and see if you guys have any questions. Thanks.
Well, if you have any good ideas for grants, our information is in the folder. Feel free to contact us at any time.